Welcome to those who've joined. We're just going to wait a moment to let the rest of the people in the waiting room join us, and then we'll make a start. Okay, welcome everybody. My name's Sonia Chaudhry. I'm Chief Executive of Action for ME and one of the members of the Partnership Board for the Genetics Centre of Excellence. Thank you so much for joining us today. We're really delighted to be able to share with you some updates about the work that we're doing. We are recording this. Participants can't be seen on the screen, so please feel reassured that nobody will see you whilst you're watching. But what we wanted to do was make sure that people who aren't able to attend today can access this. So we will share it on the Action for ME website in a couple of weeks' time. One of my comms colleagues who's going to work on the film and put it up on the website is actually on leave at the moment. So there will be a slight delay, but it will go live, and we'll let you know through the Action for ME website news, but also on social media as well. So I'll just say a little bit about the format of the session. I'm going to give you just a very brief introduction about the ME Genetics Centre of Excellence. Then we'll hear from Chris about some work that we've been doing on an MRC application. And because this is all about building partnerships, we've invited Simon Carding, who's been working with us on the MRC application, to talk about his research. We're going to hear from Audrey about her ME research as the first fellowship that's being funded through the Centre of Excellence. And then we will have time for questions and answers. And just a little reminder that if you do have a question that you would like to ask us that you've not yet sent through, there is a function through the Q&A for you to be able to type in your question. We will keep an eye on those and we will try and get through as many questions as possible. And we will be joined by the others on the screen. We'll do introductions in a little while so that you've got a whole group of us to be able to respond to the questions. So please put your questions in at any time during the presentations. So I'm going to start by just saying a little bit about the Genetics Centre of Excellence. This idea came from a conversation that Chris and I had about how we can build on Decode ME, but also how we can tackle this issue of not having enough research funding, but also not having enough genetic researchers in the field. And we can't wait for the funders to take a more strategic approach, and that is what we are all calling for to move things forward. And there are lots of others, many other charities like ME Research UK, Invest in ME, ME Association that are putting money out for research. But what we really wanted to do was to think about how we really stimulate new collaborations and bring in people from outside of the field. So this idea is still very much early in its development, but we have been doing a few things since we announced the launch of it. We held a symposium, which surprisingly is 2 years ago. That was at Edinburgh University. We had around 50 people attend and a series of presentations, and those can be viewed on the Action for ME YouTube account as well. So you can go back and look at those should you wish to. But it gave us an opportunity to really sort of develop new relationships. And one of those that was in its infancy at that point was with Precision Life. And since then, Chris and myself and our teams have been working with Precision Life on the low COMI study. That's very much focused on ME and long COVID and the genetics and looking at diagnostics. And we will be talking to you more about that through a webinar with them in due course. We have also been very much focused on getting some funding in place. There is no funding for this virtual Centre of Excellence. Action for ME has put money in to help fund the patient and public involvement. So we do recompense people for their time. And we've also put funding in for the Claire Francis Research Fellowship, and you'll hear more about that from Audrey in a moment. That's currently funded for 2 years, and we'd ideally like to be able to fund that for a 3rd year. But that's the only funding that we have that's gone into the Centre of Excellence. And Chris has given up his time as part of his role, but actually in a voluntary capacity to work as part of part of this project. The other thing that we have done is set up a partnership board, and as many of you will know through Decode ME, but also the LOCOME study, PPI, or patient and public involvement, is at the heart of all of our research work and creates huge scientific value. And so setting up the partnership board was really important to help us steer through the work that we want to do, to set up our strategy, determine exactly what it is we're going to do. And you can see some of our partnership board members on the screen at the moment. And I have a little slide which I will share with you later that shows you all of the partnership board members. I know some of them are in the audience as well today. As I mentioned, we have been working on an MRC, Medical Research Council, strategic partnership grant application to bring some money in to support the work that we want to do. And Chris will tell you about that in a second. We are now on our second submission. The first one, there were a number of things that they came back to us and asked us to consider. And one of those was to really start building some of the partnerships that we referenced in the application. And we have been successful in doing that. We are also working on some other partnerships. They're still in their early stages, and so they're not things that we can announce or share with you at the moment, but we do hope over the coming months that we will be able to share more details about those partnerships, and we will do that as soon as we're able to. So I'm now going to hand over to Chris, who's going to tell you a little bit more about the strategic partnership grant application. Thank you, Chris.
Excellent. Good. So I assume you can hear me. I've lost sight of you all, but welcome to everyone. So I'm going to tell you about this MRC partnership grant application, which we're calling PRIME, and it's building infrastructure for patients, researchers, and industry for ME, which is where the PRIME acronym comes from. And the emphasis here is on infrastructure. We believe that there is insufficient infrastructure for ME research, and I'll come back to that in a minute. So the notion really comes from this. Imagine there's a researcher, perhaps even from outside the ME research field, who has a great idea. They want to work on ME, they have a great idea, and they think it'll work. So what do they need to actually deliver their idea? And first of all, they might need a lot of people, a lot of cases and controls for their study. So they need to recruit people. Then, because of the national standards, they will need to have PPI, patient and public involvement. They will need to go through ethics procedures, often related to the recruitment of cases and controls. And they will need perhaps to make further collaborations with, say, industry or other researchers who with different expertise. So these are the barriers, really. They are also the opportunities, but these are the barriers to people entering the ME research field. They often don't know how to do PPI, how to get in touch with people interested in doing PPI. Recruitment of individuals for case control studies is very costly. And then probably will need advice from people who've been in the field for a while. So this is where we started. We think that we can build the infrastructure to allow newcomers and people who've already been in the field for quite a long time with the infrastructure, with the ability to access people who've already taken part in Decode ME for ME cases, to have a PPI pool to allow people to easily set up PPI for their structures and applications, and advice over ethics and collaborations, including with industry. So this will then meet what the key problems are that the delivery plan said, that there are key problems surrounding low capacity and capability to respond to research needs, low awareness of needed research in ME, and low levels of funded research. So we see this as infrastructural. It's not an application for funding to address a question, a hypothesis. It's not that at all. It's trying to put in a foundation upon which ourselves, and more importantly, others, can build. So our aims are the following. We aim to forge this number, 15 or more collaborations with industry or other scientists who can take the advantage of pre-existing bioresources such as Decode-ME. And we think it's ambitious, but others think that we should do more, but we'll do as much as we can to catalyze further funding for investigations into biomarkers and ME disease mechanisms and studies. We want to build bridges to many others who are working around the world, in particular in two aspects. First of all, the genetics, building on what is already ongoing, and then omics. And by that, I mean the study of proteins and metabolites often in blood, and that has been done in a variety of different places, but I think would benefit from a more joined up approach, and for the ability to people to compare and contrast their analyses in these meta-analyses across different parts of the world, including different subpopulations of the human population. And by linking to these different research projects and data, we should improve and accelerate research. Now, last but by no means least, what should come first is PPI. So if we were, as we intend to have, over 100 PPI volunteer participants ready to go for different research projects, we can make with researchers between what they require and what PPI would be interested in. So to help raise the quality and quantity of research projects. We think this is needed. At the moment, it's just done on an ad hoc basis, and we need studies that adhere to the national standards for PPI. So who are we? Who are the applicants for this MRC Partnership Grants application? Well, here in the University of Edinburgh, partnering with Action for ME, partnering with many other institutions and universities across the UK, in particular with Simon, who will be talking in a minute, and with researchers across different UK universities. We are focusing today on UK networks because that's what our funders are primarily interested in, but we will not overlook the potential for making links abroad because that's where the advances, the leaps forward will come from. So as Sonia has said, we have submitted this application. We will hear back probably around about March, April, if I remember that correctly. But what I don't want to do is to set an expectation that this will be funded. We wrote this grant in full understanding that only approximately 1 in 6 of these applications are successfully in their funding result. So nevertheless, we have tried, we've put this together. We think it's a very strong application. And essentially, we are now asking the MRC to come through with what they've always said to us, in that the ME field should put forward a partnership grant application, or more than one. And here's the first one. Thank you.
Thank you, Chris. So I'm now going to hand over to Professor Simon Carding from the Norwich Medical School, who's going to tell us a bit about his research and partnerships, and also his ambitions for ME research.
Sonia, can you hear me? You see my slides? Great, thank you. So yeah, so I apologize if this is going to be a bit of a speed rush, but I've got a lot to cover in little time. But as Sonia said, the slides will be on the charity's website to look at in more detail at your leisure. So let me start by saying that over the last 7 or 8 years, we've been working with the charity Invest in ME to develop a Centre of Excellence, for ME in Norwich at the Quadram Institute. The overarching goal of this is to establish new ways of treating the disease by uncovering mechanisms and to use those to develop evidence-based interventions. Although we primarily focus on research, that's not the only thing we do. And this is, in a snapshot, the research we do. It's infection immunity in a broad sense, specific interest in viruses and the viromes, particularly in the gut virome, autoimmunity, immunodeficiency, and increasingly getting interested in immune senescence and accelerated immune aging as a central defect underlying abnormality in ME. And then we do have translational research. We have 2 clinical trials. One is ongoing, photobiomodulation, and one that's planned for early next year. We are very outward looking. I mean, for the reasons that Chris has articulated, there are more established programs in Europe. So we are very outward looking. We collaborate a lot with Europe, and I'll talk about the organizations that have come together to help bring together all these interesting research across Europe, interested in ME. And also we have extensive interactions with the NIH in the USA. And we also have a strong advocacy feature to our activities here, which we're constantly talking to MPs in the UK government, but we also talk to funders in the US and increasingly more in Europe as well. So we believe we are strong advocates for trying to get more research done in the UK for ME. These are some of the questions that we're particularly interested with our fundamental research. What is the makeup of the virome? What viruses are present in ME patients in the gut, but also in other body sites? Are they different, distinctive from those in healthy individuals? Do they change with disease episodes, severity of disease? And how do they interact with the immune system? What is the basis of their ability to stimulate the immune system to accelerate what we believe is immune senescence? And most of our studies work with severely affected patients, which has its own logistical issues, but we primarily focus on the most severe patients. We've had 5 PhD students funded through Invest in ME and the University of East Anglia collaboratively. They've all worked extensively with Amalak Bansal, who's a GP specialist in ME, and also Tom Wildman, who's a molecular virologist at the University of East Anglia. We've published methodology papers, we've published opinion articles, and we've recently been publishing studies undertaken to look at the virus and the immune system and the interaction of the both in ME CFS patients. So we've had some productive PhD students. We've also now got 2 fellows established at Quadram. One is the Ian Gibson Fellow, which is Catherine Seaton, who was a PhD student. She's also chair of Young Emerge, which I'll talk about at the end. And the Luna Nova Fellow is Cristiano Pereira, who's just joined us and is looking more at virus-driven immune senescence in ME. And we have some contributions to the funding from American charities Solve ME and the Ramsey Research Grant Proposal. So clinical trials are an important part of what we do. We're not just doing research to uncover mechanisms. We want to use that information to develop trials. As Chris has said, one of the important things is having a PPI involvement. At Quadram, we have an established general PPI, which volunteers can sign up for to be part of any human study carried out at the Quadram Institute. And since September, we've had 50 ME patients register as a research volunteer. Then in 2023, we formed an ME CFS PPI group, co-chaired by an ME patient and a QIB team member. And they've helped us design the trials that are ongoing and planned as well. And in particular, they've highlighted issues that we wouldn't necessarily be too cognizant of, in particular access and the impact of just participating will have on their functional capacity. So the PPI has proved to be extremely important in developing a lot of our research and in particular the clinical trials. One other thing that we've done is we've developed tools, new tools. This is a patient-based questionnaire called FUNCAP to accurately assess functional capacity, which we will propose to use in clinical trials to assess the efficacy of an intervention on functional capacity. It was published early last year and it's already being used in 2 trials that we know of currently one in Harvard and one in San Francisco, I believe, are using FunCap as part of the clinical trials. Another recent activity came about from the last colloquium that was held in June this year, when a lot of clinicians and researchers got together to try and develop a consensus criteria for undertaking clinical trials. And in particular, we're trying to develop what's called an outcome index value or measure, which has been used extensively in rheumatoid arthritis clinical trials to accurately assess the impact of an intervention. And we're hoping that if we reach this consensus, then clinical trials, wherever they are undertaken, will adopt the same measures as part of their trial. Things we're looking at currently are the core measures, objective versus subjective, secondary measures, and also categorizing patients to put the right patient in the right trial to get the maximum benefit from the intervention. And I chair this with a clinician based in Denmark, and the US arm of this is chaired by Nancy Climax and Ian Lipkin. So far, we've had 3 meetings. We have another one planned in November. This is the photobiomodulation trial that's currently ongoing at the Quadram. It assesses the potential safety and efficacy of low light therapy, which has been used extensively to treat wounds and bone healing process. So it works on the principle of boosting ATP production from mitochondria. So we're currently in the process of assessing the safety and efficacy of this red light therapy in patients. We hope to report back the outcome of that early in the new year. The other clinical trial that will happen in 2025 is a phase 2b clinical trial to assess the impact of fecal microbiota transplantation to restore a healthy microbiome in ME patients with an impact on their overall physical and mental health. This is a snapshot of it. It involves a lot of people. It's taken a lot of logistics to bring all these people together so we can maximize the benefit of the trial. It will utilize Quadram's MHRA licensed FMT facility. We'll be manufacturing capsules. So we're hoping that patients that can't come into the facility will also benefit from the trial in having capsules delivered to them. And we're incorporating various objective outcome measures, assessing physical activity, functional capacity, sleep, cognitive function using neuron, and brain imaging as well in a subset of patients. So that is planned for probably happening around Easter next year. And then regarding influencing policymakers to try and get more money for ME research, we've been working with the UK government, Department of Health and Social Care, as some other people on the call have, trying to lobby for research funding. Similarly, I've been working with the NIH to have a similar lobbying there to get more funding, which has been more successful. There are now a lot more money going to ME research in the US. And we also bring in MPs locally to visit Quadram. We get them to raise questions in Parliament, and we've been to the House of Lords to generate interest at that level. So we're quite active on that front, and we work in close partnership with our communications department at the Quadram. And then coordinating, promoting international ME research. So this involves EMEA, the European ME Alliance. It's a patient organization. Membership is 18 countries. It's just received WHO accreditation. Emerge, which is for researchers. This involves around 40 researchers spread across 12 countries, which I co-chair. And then there's the EMEC, which is the European ME Clinicians Network, of bringing together people to develop best practice in clinical practice. And then Young Emerge, which is for the benefit of young and early career researchers to promote their careers in ME research. So this is currently some of the activities that Emerge is involved in, working in partnership with EMEC and EMA. So we have contributed to the Common Data Elements project that was sponsored by the NHS. We've collaborated with co-investigators through Emerge. We put in a Horizon 2020 multi-center innovation program, which unfortunately wasn't funded, but we are working on a Marie Curie doctoral network application, which will be submitted next month. We're developing new tools that can be used diagnostically and interventions. FunCap, an example of that. Clinical colleagues are also updating primers on ME/CFS for clinical practitioners that they can use in daily practice. And Emerge organizes the annual ME colloquium and we're in the 14th year. The next one will be near the end of May next year. And then finally, Young Emerge. So this came out of Emerge, and we sort of felt we had a critical mass of PhD students, fellows. We could form this across Europe. And it's to support early career researchers and encourage them to develop their career in ME. So they contribute to new research in ME through publishing original research, and I've given you examples of that. But they've also organized annual workshops for early career researchers. One was in the USA, hosted by the NIH, and then one this year was hosted in the UK, and we had about 40 students present there. So that is, I think, developing quite nicely. And one output so far, from Young Emerge is this review of clinical trials in ME and what the future perspectives are for developing new trials. So I apologize, that was a little bit rushed, but hopefully you'll be able to read more on the slides. And of course, I'm happy to answer any questions. Thank you very much.
Thank you, Simon. That was fantastic. And as rushed as it was really informative. And we can see that from some of the questions that are coming in. And I think one of the things to highlight for us with the strategic partnership grant application is, although as a group we're very much focused on the genetics, we have to collaborate as a field, as a group of researchers, charities and others. The collaboration is so important, and we can't just focus on one field. We have to look across the spectrum. So thank you so much, Simon, for sharing more about your work. I'm going to come back to you with some questions afterwards, but before then,
sure.
We'll hear from Audrey, who is the Claire Francis Research Fellow working at Edinburgh University. Thank you, Audrey.
Thank you, Sonia. First of all, can you see my slides? Can you hear me? And can you see my cursor?
Yes, all good.
Okay, excellent. All right. Thank you so much for the opportunity to present. My name is Dr. Audrey Ryback, and today I'm going to tell you about the work we've been doing to answer the question, do ME blood factors change the behavior of cells? So as Sonia's already mentioned, I am the first Claire Francis Research Fellow funded by Action for ME. My fellowship started just over a year ago, and I'm based at the ME Genetics Center of Excellence. The overarching hypothesis of my fellowship is that the behavior of cells grown in the lab is different when they're exposed to ME serum or plasma than when they're exposed to serum or plasma from healthy controls. Now, this work addresses two of the ME research priorities identified by the ME Priority Setting Partnership, namely, how can an accurate and reliable diagnostic test be developed for ME? And priority number 10, how are mitochondria affected in ME? Right, so before I tell you about what we've been doing in the lab, I'm just going to give a little bit of context. So you'll probably remember from school that the mitochondria are the powerhouses of the cell. They can produce energy in the form of ATP. And the process of generating ATP requires oxygen. So I've highlighted the oxygen here with the red arrow. And so we can test mitochondrial function by measuring how much oxygen cells are consuming. Now, a common thing to do in the laboratory to test this is to perform a mitochondrial stress test where different parts of this ATP generation machinery are interfered with toxic compounds. And as a result, you get a change in oxygen consumption rate. So this cartoon just illustrates what these kinds of traces look like. So along the y-axis, we have oxygen consumption rate. Along the x-axis, we have time. And then as these different toxic compounds are added, you see a change in oxygen consumption rate, which corresponds to different aspects of mitochondrial function. So the first aim of my fellowship is actually to replicate a previously published finding by a Norwegian group. This was an experiment done in a paper by Fluge et al. In 2016, where they performed such a mitochondrial stress test, and they saw that under conditions of aerobic strain, the maximal respiratory capacity of cells that had been exposed to ME serum was higher than cells that had been exposed to healthy control serum. So this corresponds to this green area here. And they saw differences at other points as well, but the reason we were interested in this is that the magnitude of that difference is quite large. So you might be wondering why I want to replicate a study that has already been published. And the reason for that is illustrated in the graph on the left. So this is a survey published in Nature where they asked researchers across different scientific fields whether they'd ever experienced the failure to reproduce an experiment. And in biology, nearly 80% of the researchers had experienced the failure to replicate someone else's experiment. And this doesn't mean that the original papers or researchers had done a bad job. This just reflects the fact that we tend to see novel results in publications, things that are novel and surprising, and oftentimes upon replication, they don't hold up. So we need to see the same result from multiple groups or more than once across the field to have confidence that a finding is true. Now, in our case, several small studies suggest that cells behave differently when they're exposed to serum or plasma from ME patients compared to serum or plasma from healthy controls. But in order to really be convinced that this is the case, we need a successful replication of one of these experiments. The previous studies have all used slightly different methods or different types of cells. So a successful replication would give us and funders the confidence that this is a research avenue really worth pursuing. And eventually, this could hopefully lead us to identifying a diagnostic marker or something about the biological processes in ME. So what I've been doing in the lab so far is to grow healthy muscle cells in the lab and then expose them to serum either from ME patients or healthy controls, and then perform one of these mitochondrial stress tests I described earlier on this machine here. And we seed, you can see here the 96 well plates in which we seed the cells. And if we zoom in on one of these wells, you can see the individual cells nestled up against one another, covering the bottom layer of the well. And this is where we then measure the oxygen consumption rate. And I can tell you, so this is an image of one of the wells that we've tested over the course of the experiment, but I don't actually know whether these cells have been exposed to serum from ME patients or from healthy controls because we have blinded the experiment, which means that we won't reveal which group a given participant's serum belongs to until we've finalized our analysis plan. And this is so that we don't bias the results. So what we've done so far is to collect a cohort in Sheffield of 67 people with ME and 53 healthy controls. This was done in collaboration with Caroline Dalton. All of our participants are female. All of our ME patients meet the CCC or the IOM diagnostic criteria. Each participant's serum was used to treat muscle cells in culture 5 wells at a time across 3 separate experiments. So that gives us 15 technical replicates. So we've generated a total of 1,800 traces across 21 experiments. And the graph below is just one of those experiments to show you some example data. So again, along the y-axis, we have our oxygen consumption rate, which we measure. And then along the x-axis, our time points. And as we add these different mitochondrial toxins, we see a change in oxygen consumption rate. And each of these traces corresponds to the oxygen consumption rate measured in a single well, like the one I just showed you. And each of the colors corresponds to 1 participant. So we run 18 participants on the plate, and there's a mixture of ME patients and healthy controls on each plate, but we don't know who is who at this stage. Right, so all of this, we've we want to get an answer that is as definitive as possible to our question. And so there are a few things that we've done to try and achieve this. The first thing is that we've used a large sample size. So the original study had 12 ME patients and 12 healthy controls. We have 67 patients and 53 controls. We're trying to understand and correct for sources of noise or variation in our data. So things that are due to technical effects or how different cell numbers might be affecting the results and sample batches. And we want to do all of that and finalize our analysis plan before we run our final analysis and reveal the result. And crucially, we've also pre-registered our study, which means that we have defined in advance what we're measuring and how we intend to analyze it. And the purpose of this is to be really transparent about what our hypotheses are, so that when we do get our results, we're not then tempted to try and spin a different story from that. So it provides a level of transparency about the research process. So what do we do next? The next experiment that is planned is to use cell painting, which stains 6 different compartments of the cell with different colors and then with different dyes, and then image them. And what this allows us to do is look at how serum affects the cells in terms of the shape and size of different organelles. So we'll look at changes to the shape and size of the mitochondria, but also other organelles within the cell or their location or their proximity to one another. And this could generate some hypotheses about how serum is affecting the cells beyond just how it affects the mitochondria. So the take-home message is that we're trying to answer the question, do ME blood factors change the behavior of cells? And in order to get an answer that is as definitive as possible, we're performing first a replication. We are using large sample sizes, we've blinded our experiments, and we're trying to account for technical noise as best we can. And if we do succeed in replicating this result, we would then want to ask the next question, which is, what is in the serum that causes these changes? And that might be and those factors in the serum might be suitable to be developed as a diagnostic or tell us something about the biological process of ME, but it would provide a really solid evidence base from which to start asking these further questions. So finally, I just want to thank everyone who's been involved in this. There have been lots of people who have helped, in particular, Charlie Hillier, who is a patient with ME, who's co-producing this research with me, Professor Chris Ponting, and Caroline Dalton and her team at Sheffield Hallam University. Thank you.
Thank you, Audrey. So we're now going to move into the question and answer session. And I've got a number of people who have joined me on the panel. What I'm going to do is ask people to introduce themselves as I direct questions to you rather than doing around all together. So I'm actually going to start with our PPI partners, and that's Kelly and Helen, just combined your name there. And maybe you could just start by saying your name and what you do and what your role is with the partnership board.
I'm Helen Baxter. Some of you will know me from 25% ME Group, but my role here is to ensure that research is accessible for people with severe ME, and they can find themselves often excluded from that research. And we did actually acknowledge in our terms of reference that researchers must be encouraged to make considerations for people with severe ME. And I've also considered the difficulties people with severe ME have when, say, they look at the screen, and that's influencing the design of our website. And I think activities in the future will involve people on the PPI working with researchers directly and providing information in a way that is accessible, particularly to people with severe ME.
Thank you, Helen. Kelly.
Hi, I'm Kelly McClellan. I've had ME for 30 years now. My role on the board I see is basically to provide a voice from the patient perspective to ensure that things that the community would be interested in are being done. I think it's also important to communicate with the community and get feedback from them. So it's not just my opinion that I'm representing, it's other people with ME.
Great, thank you, Kelly. And Hayley, maybe you could introduce yourself. You're our newest member to the partnership board and you're involved in research yourself. So maybe you could just say a little bit, just a snapshot of your research and also what attracted you to the partnership board.
Yes, I am Hayley. I'm a PhD candidate at Stellenbosch University, and I'm busy focusing on research into ME and long COVID. Particularly focusing on the coagulation aspect of the conditions. And what really drew me to the board, I think, is just the uniqueness of it. I think that being a sort of community or like a platform that involves such different perspectives, I think is very unique. And I think that it just shows the importance of making sure that the research around ME is founded on research as well as lived experiences. So I thought that was really great about the board.
Okay, thank you, Hayley. And Doug, welcome. You've just recently joined us. I'd like to direct a question to you, if that's okay. So maybe you could just introduce yourself, but also maybe you could tell us about what attracted you to becoming part of the MRC Strategic Partnership Grant.
So yeah, I'm Doug Kell, and I've been working with Risha, who Hayley also works with, for something north of a dozen years now, on initially iron metabolism, and then we kind of discovered the anomalous clotting that turns out to be amyloid in nature, and important in a variety of chronic inflammatory diseases, and showed that the amount of anomalous microclots in ME/CFS patients was something over 10-fold greater in them than in the equivalent healthy controls. So obviously, from a scientific point of view, we want to keep this going. From a human point of view, I've come to meet many long haulers and ME/CFS people and would very much like to help them to the extent that a non-clinician can. And that's why I was keen to join the party, really. Nothing much more than that.
Great, thank you, Doug. And Simon, we've got a number of questions in the Q&A function around how people can get involved in the work that you're doing, the trials. Is there a website that you can point people to, email, or can you give information about when you might be recruiting if you're not already?
Yeah, so to become a QIB research volunteer, that can be found via the QIB website. There's a form you fill in, download, and then mail in to individuals that will then put you on the register. And depending on the nature of the trial, you'll be approached to ask if you're interested in participating. Now, our ME, upcoming ME clinical trials, the recruitment is via the East Coast Community Healthcare CFS facility based in Lowestoft, Suffolk. So patients have to be registered with them in order to be considered for the clinical trial. Currently, they have around 1,700 patients registered with them, and there are approximately 300 individuals that have already expressed an interest in the upcoming clinical trial. But that is the way to get into the next clinical trial. But if you want to volunteer for lots of studies, then that can be done by the QIB website, volunteer page.
Great, thank you, Simon. And Audrey, one for you. You're obviously the first Claire Francis Research Fellow funded by Action for ME. How has this type of charity funding shaped your work?
Thanks, Sonia. Yeah, I think the biggest way in which it's shaped my work is that my research interests and Action for ME's interests are perfectly aligned. So we want to have the biggest impact we can on ME research and deliver research that will drive things forward. And it can be quite hard to make an argument for example, replication studies to funders, because it's not particularly attractive, but it is so important, especially in ME research. So Action for ME understand that, and we've been working in partnership, and we really, you know, want to make the biggest impact we can in the time and funding that we have.
Great, thank you, Audrey. And I've got two more questions I'm going to ask you whilst I'm with you. The first one is, what's the theory behind why the mitochondria become damaged?
Right, so we don't actually know whether the mitochondria are damaged in ME. I'm presuming that this question is in reference to mitochondria in ME, but you know, the model that we're using, we're taking healthy cells and we're exposing them to the serum, to the liquid portion of the blood from patients. So it is quite an artificial system. But the purpose of doing these experiments is to have a starting point from which we can build onto further research and find out what is it in the serum that is causing these changes, and might that tell us something about the biology of ME. So we don't know at this stage, unfortunately, whether mitochondria are damaged in ME or yeah, anything related to that, but it's really finding a starting point for that research.
Great. And then the last one, somebody said it's great to hear about your research. When are the results going to be out?
Yes, so we have just finished data collection of the experimental work, and at the moment we're checking the quality of that data to make sure that that we have everything we need, that we don't need to repeat any more experiments. And once we have that, we'll finalize our analysis plan, and then we intend to publish our results as a preprint, so they will be out as soon as they can, but we need to make sure that everything is done to the best of our ability and we have the most robust answer we can give. So unfortunately, I can't give a specific timeline or date for that, but we are trying, as we do appreciate that we need answers soon, and we're trying our best to do that as quickly as possible.
Great, thank you, Audrey. Chris, I'm going to come to you. We've had a number of questions from people saying, how do I sign up for the PPI stuff? I know Simon's answered that in terms of the work he's doing, where we talked about the strategic partnership application. We don't have funding yet, so you know, if we're successful, we will certainly let people know how to sign up there. But maybe, Chris, you can tell us a little bit more about what we might do if we're not successful. And the question was very much around building on Decode ME and ME genetic research, but I'd like to specifically pick up the three areas that you identified in the MRC strategic partnership grant application, and what conversations have we had as a partnership board about how we might be able to take that forward.
Thank you. So we will always do what we can, given limited resources, if not funded. We will always look to forge new collaborations in the UK and abroad. And I think we've been successful at that, and in future meetings we'll be talking more about that. But it's a real struggle. We have limited time, energy, people, and it isn't just money, but it's about what is funded. And if what is funded in Prime then allows us to forge these new collaborations and to bring in PPI pool to make new discoveries indirectly, or to facilitate others to making new discoveries, that would be just a fantastic use of taxpayers' money, in my view. What are we going to do otherwise? Well, we're just going to carry on working as hard as we can, as best we can, but we're not accelerating research. And to me, this is what Prime would do if funded, is to accelerate research towards what everyone wants, which are new therapies.
Great, thank you, Chris. And obviously we've got lots going on with the Decode ME project at the moment, where the webinar today isn't specifically about Decode ME. So any questions that we've had around Decode ME, we will be able to pick up when we have our next Decode ME webinar. And the same with Audrey. We'll be very keen to get results out as soon as possible, but there's a lot of work we've got to do to get us to that point. So Simon, a couple of questions from you. And first of all, somebody said how great it is to see researchers in Edinburgh and Norwich doing something together, and more collaboration, please. And I think you've heard from everybody today that that is absolutely at the heart of what we're wanting to do. So first questions is Young Emerge focused on only physiological research, or does that include psychiatric too?
It's open to anybody that would be considered an early career researcher, irrespective of their specialist discipline. We are trying to be inclusive, so it's open to anybody and everybody that has undertaken research in any aspect of ME or other chronic virus infection related disease that is interested in becoming more ME research focused themselves. So it's by no means exclusive.
Great, thank you. And another question about the link between ME and the microbiome. Where did that theory come from and why? And I think I heard you say fecal transplant, and you were talking about capsules will be used in the research trial. So maybe just, you know, where the theory came from and why it's there and why you're looking at it.
Yeah, so I think it originally stems from patient observations, because the very large proportion of ME patients have GI symptoms, and for some of those patients, a large number of them, that seems to precede the onset of developing ME. That was followed by some initial studies carried out by groups in Belgium and Australia identifying changes in specific types of microbes in the gut. And then an individual called James Borody in Sydney started to use FMT to treat ME patients, and he published in, I think, 2012, claiming that there were some quite striking beneficial effects of his FMT, in that some patients were symptom-free after 15 years of having an FMT therapy. So we wanted to take that and put it in a clinical trial setting to properly assess the efficacy of FMT, and that's what we've been working towards for the last 2 or 3 years. And since then, other publications have come out on large numbers of patients showing that they have alterations in their bacterial component of their gut microbes. We've published on alterations in the virus populations of the virus. So there's mounting evidence for changes. And of course, the key question is that cause or effect? So a clinical trial setting would allow us at least to take a step towards addressing that question.
Great, thank you, Simon. And somebody's asked about the link that you shared on your slide for the volunteers. So maybe Daphne or Alice, who are colleagues working in the background, maybe you could just post that in the Q&A function so that our participants can see that. That would be really helpful. Thank you. There's a question here. It's a comment, but I'm gonna turn it into a bit of a question for our scientists on the panel, so you can maybe flag if you'd like to take this one. I see studies happening for both ME/CFS or for fibromyalgia, and they seem to use 20 to 40 patients. This goes back to the numbers and how statistically viable, powerful studies are. They find something interesting, and then we don't hear anything more from the group thereafter, and they've shared one. That's never going to be enough evidence to reach changes in the clinic. Have you got any comment about that, or views around what we need to be doing differently? Doug's come on screen, so I will let him take this first.
Well, I've just looked at the paper on my screen to the left, which is about microRNAs, and obviously there's lots of them, more microRNAs than the number of patients, and unsurprisingly, they found a difference. And so why it didn't get followed up was either because actually there wasn't a difference, or they didn't get funding. We don't know which of those two it was, so I suspect the first of those, to be honest. And those who followed John Ioannidis's many writings on the subject will find that very unsurprising, that underpowered studies do not withstand scrutiny.
Any other comments from our scientists or PPI colleagues on the panel?
I think I'm on record as saying only believe something if you've seen it twice in different groups and well-designed studies. It's echoing what Doug has just said. It is awful that there are so many studies that are underpowered, that are being funded and then published, which almost always at the end say, oh yes, well, this is these might be interesting, but we need further studies to determine whether this is true or not. Well, perhaps we as scientists should build in replication into our studies in the first place, rather than giving false hope to people.
Thank you, Chris. And I can say we were discussing that very thing at the management group meeting for Decode ME this morning. Anyone else before I move into the next question?
I think we absolutely need to try and increase sample sizing, but in order to replicate those pilot studies, funding really does need to be available to researchers.
Yeah, absolutely.
It's also the entire reason for Audrey's work is replication. So I think we as a community are very much aware that of exactly what Chris was saying, that if it's not big enough, it's not really, it's meaningful. It's interesting, but it's not meaningful yet.
Great. As always, we have loads more questions than we have time for. So we've had a series of questions about how people can find out about joining PPI, about trials that are happening. Chris.
Yes, well, hopefully we'll be funded so we can set up the PPI pool. But I did want to mention the NIHR's website, Be Part of Research, in one word, at the NIHR, which is for people in England, Northern Ireland, Scotland, Wales, where people can register for a study that's right for you, they say. And that includes people with ME, long COVID, etc.
Great, thank you, Chris. Also, Action for ME on our website, if we get contacted by researchers, we do put research that up on the page where people are looking for recruits. We don't vet that research, we don't offer comment on it. So if there are researchers out there that are trying to recruit people, we can share that. And it is something we can take away and think about through the Centre of Excellence as well. There's some feedback about the FunCap scoring and how excellent it is, and hope that it will get into GP consultants' rooms. I've heard that. Yeah, I heard that on a number of occasions outside of today. We've also got a question around how we can help in the future. So there are lots of ways you might want to participate in studies. Many of the charities, Action for ME, Invest in ME, the others do fund research, like Audrey and others that Simon has shared today. You can help spread the word. We're in the process of setting up the all-party parliamentary group. We need MPs to sign up as members and officers. So, visit the charities' websites. There are a number of actions that people can take. We also recognise that there are many that are unable to use their energy for things like that. So we don't want anybody to feel pressured in any way. And then a suggestion from Michael, hi Michael, who suggests that Crunch ME might be useful in calibrating people new to the field. So if you haven't seen that already, it's a fantastic resource, and that is crunchme.org, and it's lots of information around ME research. So definitely worth a look if you haven't already. And then there's a question around plans to work with the ME Biobank in London. A number of studies are accessing the samples that are available through the biobank. I think Audrey, you did, wasn't? Yes, you did too, as well in your research. At the moment with the MRC application, they're not part of the strategic partnership grant application, but the whole purpose of us working together, Genetics Centre of Excellence, is not purely to focus on genetics. It's about building those relationships, and we welcome partnerships with everybody. So that brings us to the end of the webinar. What I would like to do first of all is, as I share my screen, I just want to thank the other members of the partnership board who are in the audience with us today, some of them, so you should be able to see that on your screen at the moment. We have a number of members as part of that. I'd like to thank our panelists and our presenters today, and Daphne and Alice, who have been working in the background, pulling all of this together, keeping us on track. You might want to turn your cameras on if you want to say hello. Alice is the Decode ME comms officer that works with Action for ME. Hi, Alice.
Hi.
And
Thanks for joining, everyone.
Daphne is Action for ME's research manager. So thank you both for all your work in the background. And thank you to everybody that's joined us today. We will be launching our new website soon. We will be able to pick up questions and answers and lots more information on there. And we'll let you know through social media and other mechanisms when that happens. Thank you very much, everybody. Goodbye.